6%. That is the amount of archaic human DNA humming inside the cells of a living person in the highlands of Papua New Guinea right now. Not 0 point something. Not a faint echo washed out across 40,000 years. 6% of their genome is not human at all in the sense you were taught in school. It belongs to a ghost species, a branch of the human family tree so deeply buried that for most of the 20th century, no one even knew to look for it. In a typical classroom anywhere in the world, the human story gets drawn as a clean line. Apes to Australopithecines, to Homo erectus, to Neanderthals, to us. That line is wrong, not slightly incomplete. Stop and think about what 6% actually means. 1 out of every 16 nucleotides in the DNA of a Papua New Guinean Highlander does not trace back to Homo sapiens. It traces back to a population we have never seen a complete skeleton of. A population whose entire fossil record fits in a shoebox. A population modern science did not confirm existed until 2010. And not just one population. The latest research, published just a few years ago by a team from Cambridge, revealed something even stranger. Papuans carry the DNA of at least 2 entirely different groups of these archaic humans. 2 groups that had been separated from each other for nearly 300,000 years before either of them met the ancestors of the people living in the highlands today. The textbook picture of isolated Stone Age tribes perched at the edge of human history is not just outdated, it is backwards. Papua is not the periphery of the human odyssey. Genetically speaking, it is the library, the last living archive of a lost branch of humanity that vanished everywhere else on the planet. Sit with that number, 6%, and the deeper finding in that Cambridge study was even more specific and more destabilizing than most people realize. We will get to it, because it changes the entire shape of what we thought we knew about the first modern humans who walked out of Africa. But to understand why 6% hits like a thunderclap, you first have to understand the old story, the one that held for most of a century and a half, the textbook version of the peopling of the Pacific that placed Papuans at the very bottom of a racial and civilizational hierarchy, a story that colonial anthropology built brick by brick, and that DNA has now dismantled brick by brick. In the 19th century and well into the 20th, Western anthropologists looked at the highlands of New Guinea and saw what they wanted to see, a living museum of the Stone Age. Here were people who cultivated sweet potatoes in mountain gardens, who built no cities, who fought with bows and obsidian-tipped arrows, who spoke hundreds of mutually unintelligible languages, clustered so densely that one valley often could not understand the next. To the Victorian imagination, this was not a complex and sophisticated adaptation to a uniquely rugged landscape. It was primitiveness frozen in amber. The racial science of the era placed Melanesians and Papuans near the bottom of a supposed ladder of human development, above Aboriginal Australians only in some schemes, certainly far below the civilizations of Asia and Europe. Dark skin in the Pacific got explained as the mark of an ancient, inferior wave of migration, a first failed experiment in being human that later, more advanced peoples had swept aside everywhere except in these remote, backward corners. The old story said that modern humans arose once, in Africa, and then spread outward in a clear sequence, replacing everything that came before them. Papuans were cast as remnants of that earliest, least developed layer. This story had the advantage of sounding coherent. It made colonialism feel like natural law. If some peoples were simply less evolved, less capable, then the whole structure of empire could be recast as a kind of biological inevitability rather than a brutal political project. The maps got drawn, the racial categories hardened, and for generations school children across the Western world learned that the true human story ran from the Fertile Crescent to Greece to Rome to Europe and then outward, civilizing as it went. The highlands of New Guinea barely registered, except as a cautionary visual aid. This is what we left behind. It sounded clean. It sounded complete. Real history almost never behaves like a tidy paragraph. And what genetics began to uncover starting around 2010 would reveal the old colonial story to be not just slightly incomplete, structurally wrong. The first crack appeared in a German laboratory whose name you should know, the Max Planck Institute for Evolutionary Anthropology, in Leipzig. A soft-spoken Swedish geneticist named Svante Pääbo had spent the better part of a decade obsessing over a problem most of his colleagues considered impossible: extracting and reading ancient DNA from bones tens of thousands of years old. The DNA molecule degrades over time, shattering into shorter and shorter fragments. Contamination from modern human handlers, from bacteria, from fungi, is orders of magnitude more abundant than any traces of the original genome. Most researchers had decided the whole enterprise was a fool's errand. Pabo built an entire cleanroom protocol, a cathedral of sterility, to make it possible. In 2010, his team at Max Planck published a paper in the journal Nature that changed everything. The bone they had sequenced was a sliver of a finger, probably from a young girl, recovered from a cave called Denisova in the Altai Mountains of southern Siberia, a landscape of birch forests and cold winters thousands of miles from the tropics. The finger bone was somewhere between 50,000 and 70,000 years old. When the genetic data came off the machines, the team expected one of two familiar patterns. Either the bone belonged to a Neanderthal, whose range was already known to have stretched across Eurasia, or it belonged to one of our own ancestors, an early modern human pioneering what was then the frozen edge of the known world. What the sequencers saw was neither. The mitochondrial DNA, inherited only from the mother, diverged from both modern humans and Neanderthals roughly 1 million years ago, a split so deep it placed the Denisova individual in a previously unknown lineage. The full nuclear genome, published later, refined the picture. Denisovans were a sister group to Neanderthals. Their own population had split from the Neanderthal lineage around 400,000 years ago, while the common ancestor of both had diverged from our own Homo sapiens lineage, roughly 600,000 to 700,000 years before the present. In other words, Denisovans were a distinct branch of the human genus, as different from Neanderthals as wolves are from coyotes. And yet here they were, living in Siberia alongside Neanderthals and, eventually, alongside modern humans. The family tree had a whole limb no one had ever seen before. Let that sink in. One finger bone, the size of a pencil stub, from a cave nobody had paid much attention to for decades, and it launched an entire new species into the scientific record. But the most stunning implication of that 2010 paper was not the existence of the Denisovans themselves. It was something else the team noticed when they compared the new genome to the genomes of living human populations around the world. The Denisovan sequence did not match any modern group particularly closely, except for one region. One group carried Denisovan DNA at levels far above the faint background noise. The highest signal came from the highlands of Papua New Guinea and from the islands of Bougainville in the Solomon Sea. In 2011, a follow-up study led by David Reich, a mathematician turned geneticist at Harvard, published in the American Journal of Human Genetics, put a hard number on what the Leipzig team had glimpsed. Papuans and other Melanesians carried between 4 and 6% Denisovan ancestry in their genomes, while mainland East Asians, Europeans, and Africans carried virtually none. The ghost species was not extinct. It lived on, in the blood of living human beings, on the far side of the planet from that frozen Siberian cave. Read that number again. 4 to 6% archaic human ancestry, concentrated in one corner of the planet, undetectable in almost everyone else. The discovery threw a wrecking ball through the clean line of the old replacement model. Modern humans had not simply swept out of Africa and replaced every earlier hominin they encountered. They had met them, interacted with them, and, in at least one specific moment in deep prehistory, reproduced with them. The consequences of those encounters are written into the genomes of people living in the Papua region right now. And that was only the beginning. The genome had one more secret waiting. In 2019, a team led by Guy Jacobs, a researcher then at the University of Cambridge and now at Nanyang Technological University in Singapore, published a landmark paper in the journal Cell. Jacobs and his colleagues, including the veteran population geneticist Murray Cox, had sequenced 161 new genomes drawn from 14 different populations across island Southeast Asia and New Guinea. For the first time, geneticists had the resolution to look at Denisovan ancestry not as a single uniform block, but as a spectrum, a mosaic of different archaic contributions layered one on top of the other. What they found inside the Papuan genome was something nobody had predicted. Papuans do not carry the DNA of just one Denisovan population. They carry the DNA of at least two entirely separate Denisovan lineages. The researchers labeled them D1 and D2. These two archaic populations had diverged from each other approximately 283,000 years ago. Chew on that number for a moment. 283,000 years of separation. To put that in perspective, the distance between D1 and D2 is roughly as great as the genetic distance between modern Homo sapiens and Neanderthals. Denisovan is not the name of a single homogeneous group. It is a catch-all term for a deeply diverse branch of archaic humanity, a branch that spread across Asia, split into multiple populations, and inhabited different environments for hundreds of thousands of years, accumulating genetic differences comparable in scale to differences between entire species on other branches of the hominin tree. The ancestors of modern Papuans did not meet a single Denisovan group in a single place at a single time. They encountered at least two separate branches of this archaic radiation, in different locations, in different centuries or millennia, and their descendants carry the blended inheritance of those multiple encounters. And the Jacob study found an additional Denisovan lineage, designated D0, distinct from both D1 and D2, present at lower levels in East Asian populations. The Denisovan homeland was not a dot on the map. It was a continent-wide human landscape, and the Papuan genome is the most complete surviving record of its vanished diversity. Picture what this means. Try to imagine standing somewhere in Wallacea, the spray of islands between mainland Southeast Asia and the joined Australia-New Guinea landmass that geologists call Sahul, maybe 45,000 to 60,000 years ago. The sea levels were lower then, but not low enough to eliminate the deep water channels that run between the biogeographic zones. Modern humans, the ancestors of today's Papuans and Aboriginal Australians, were moving eastward from the Asian mainland, island hopping, building watercraft, reading currents and stars. They were already the most restless and adaptable hominin the planet had ever produced, but they were not moving through empty wilderness. As they crossed the straits and made landfall on islands that no longer exist as we knew them, they met other humans already there. These were the Denisovans, D1 and D2, populations that had arrived hundreds of thousands of years earlier, that had adapted to the humid heat and the dense forests and the unfamiliar pathogens of equatorial Southeast Asia, that had developed their own cultures, their own toolkits, their own ways of being human. The meetings happened. Gene flow occurred, the modern human groups who eventually pushed into Sahul and split into the Aboriginal Australian and Papuan populations around 37,000 years ago, as shown by a 2016 Nature study led by Anna Sapfo-Malaspinas from the University of Copenhagen, carried with them the genetic signatures of those meetings in their cells. Their descendants in the highlands still do. If your family carries roots from the New Guinea highlands or from the islands of Melanesia, the specific mixture in your DNA right now reflects a history unlike almost anywhere else on Earth. Not one archaic introgression event. At least two. Not a single ghost population. A whole radiation of them, preserved nowhere else at this concentration. This is not a footnote. This is the main text. Now bring the earlier thread back, because it connects to everything you just heard. If everything so far surprised you, the next finding turns the whole thing over. Denisovan genes in living Papuans are not just genetic noise, passive souvenirs of ancient encounters that natural selection has been slowly scrubbing away. Many of them are functional. They do things. Some of them may even help keep people alive. In 2014, a team including Emilia Huerta Sanchez, now at Brown University, published a study in Nature that cracked open the field of archaic functional genomics. The researchers were studying Tibetan Highlanders, a population famous for their ability to survive and thrive at altitudes where oxygen levels would disable or kill most other people. Tibetans carry a specific variant of a gene called EPAS1, a gene that helps regulate the body's response to low oxygen. When Huerta Sanchez and her colleagues traced the evolutionary history of that variant, it did not trace back to modern humans. Its closest match was the Denisovan genome from that Siberian cave. At some point in the deep past, the ancestors of Tibetans inherited a version of EPAS1 from Denisovan relatives, and natural selection then amplified it generation after generation because it gave carriers a survival edge in the thin air of the Tibetan plateau. The Tibetan EPAS1 story is now among the most famous examples of adaptive introgression, but it is far from the only one. In 2016, a study led by Benjamin Verneau, then at the University of Washington, published in Science, sequenced the genomes of 35 Melanesians from 3 different island populations and mapped exactly where in the genome Denisovan ancestry clustered. The paper found that Denisovan DNA was not uniformly distributed across the chromosomes. It was concentrated in specific regions, often in genes related to immunity, metabolism, and skin physiology. Natural selection had favored some archaic variants and purged others. The Denisovan genes that stuck around in Papuan and Melanesian genomes tend to involve the immune system, particularly genes that help the body detect and respond to pathogens. This makes deep evolutionary sense. The Denisovans had been living in island Southeast Asia for hundreds of thousands of years, adapting to tropical diseases, local parasites, viral ecologies that modern humans from mainland Eurasia or Africa had never encountered. When the modern human ancestors of Papuans arrived in those same tropical environments, meeting Denisovans and interbreeding with them, they picked up pre-tested immune solutions, genetic shortcuts to surviving a new disease landscape that would otherwise have cut them down. The Denisovan legacy was not just a mark of who they met, it was a survival kit. Sit with what that means. Archaic genes kept modern humans alive in a new world. The encounter was not only sexual, not only cultural, it was medical. But not every archaic gene was a gift. The Verno study also found that Denisovan ancestry was markedly depleted in certain parts of the genome, especially on the X chromosome and in genes associated with brain development. In other words, some Denisovan variants were incompatible with the modern human genetic background. They caused cases of reduced fitness, maybe infertility, maybe cognitive effects, and natural selection scrubbed them out with ruthless efficiency over the generations. The genome of a modern Papuan Highlander is not a passive archive. It is a landscape scored by conflict, cooperation, survival, and loss. Some Denisovan gifts were kept, others were thrown away. And then there is the geography. The fossil Denisovan record, as of 2025, remains almost absurdly sparse. One finger bone from Denisova Cave, a few teeth, a partial jawbone from Shahe, high on the Tibetan Plateau, published in 2019, and confirming that Denisovans lived at altitude long before modern Tibetans arrived. A skull fragment from Denisova Cave, published in 2024, which turned out to belong to a first-generation Neanderthal-Denisovan hybrid, a daughter of two worlds. That is it, an entire continent-spanning population, present in Asia for hundreds of thousands of years, known from a handful of physical fragments stored in climate-controlled cabinets. Everything else we know about Denisovans, we know from the living genomes of Papuans, Melanesians, Aboriginal Australians, Philippine Negritos, and, at lower levels, East Asians. Think about that for a second. The Denisovans are simultaneously one of the most important and least visible branches of the human family. Their existence was unsuspected. Their wide geographic range was unsuspected. Their internal diversity was unsuspected. And the single richest repository of their genetic legacy is not a frozen burial in Siberia. It is a living human community in the mountains of New Guinea, a place that colonial science once imagined held no meaningful history at all. The political and historical dimensions of this are impossible to ignore, and the script should not try to ignore them. For more than a century, Papuan people were depicted in Western anthropology and popular imagination as a kind of living fossil, a throwback to a simpler, less developed stage of humanity. The Highlanders were photographed wearing elaborate headdresses and carrying stone tools in National Geographic spreads that framed them as survivors from the dawn of time, isolated from the forward current of civilization. The racial hierarchies of the colonial era placed them near the bottom of the scale, precisely the opposite position from the one they actually occupy. The Denisovan findings do not nudge this framing, they invert it. Papuans are not the marginal survivors of an old, simple humanity. They are the central inheritors of the most complex human diversity the planet has ever produced. Their genomes preserve an entire branch of archaic humankind that was erased everywhere else on Earth. A branch whose diversity we are only beginning to fathom, whose adaptive gifts we are only beginning to trace, whose story we are only now learning to read. The people who were once cast as the end of the human line are, in genetic reality, the keepers of its richest archive. If the story stopped here, it would already be powerful enough on its own. But ancient DNA had one more secret waiting, a secret that makes the Papua genetic story even deeper, because it implies that the Denisovan encounters were not singular events, but part of a broader pattern of archaic admixture across the entire region. In 2021, a study led by Maximilian Lorena, a researcher at Uppsala University in Sweden, published in the journal Current Biology, examined the genomes of 118 individuals from ethnic groups across the Philippines. The team was looking for Denisovan ancestry. What they found shifted the geography of the Denisovan story yet again. The highest level of Denisovan ancestry in the world, slightly exceeding even the Papuan Highlanders on some measurement panels, belonged to a single small ethnic group, the Aita Magbukon, a Negrito community on the island of Luzon. The Ida Magbukan carry around 5% Denisovan ancestry, and their genome showed signatures of a Denisovan introgression event that was independent from the events that gave Papuans their archaic DNA. In other words, the ancestors of the Philippine Negritos met and admixed with Denisovans at a different time, in a different place, from the ancestors of Papuans. The map of Denisovan human interaction was not a single romantic meeting. It was a web. Modern humans moving through Wallacea, the Philippines, and insular Southeast Asia encountered different Denisovan groups on different islands in different millennia, and the genetic signatures of those encounters were preserved in the separated populations that descended from them. Papua holds the highest average across a large regional population, but the Philippine IDA finding shows that the legacies of these encounters are scattered, layered, and still only partially mapped. If you are feeling a strange mix of anger and awe right now, you are not alone. Anger because an entire century of scholarship dismissed and diminished the very communities that turn out to have been carrying the most extraordinary genetic legacy in the human story. Awe because the science itself, in barely a decade and a half, has peeled back a layer of our past that nobody even imagined existed before 2010. Leave one word in the comments capturing how this makes you feel. Just one word. Now, it is important to say plainly what the science does not prove, because the story is still being written, and honesty about the limits of our knowledge is exactly what gives the findings their credibility. The exact percentage of Denisovan ancestry in Papuans varies depending on the measurement method. Whole genome analyses and SNP-based panels do not always yield precisely the same number. Most studies cite a range of 3 to 6%, with 4 to 5% being the most common central estimate. The two Denisovan populations, D1 and D2, are inferred from statistical models of the modern genomes, not from direct fossil evidence matching each lineage to a specific skeleton from a specific archaeological site. The models are robust and have been replicated, but they remain inferences, probabilistic reconstructions of a past we cannot directly observe. And the fossil Denisovan record, as noted, remains vanishingly sparse. Most of what we know about this branch of humanity comes from computational genomics, not from dusty excavations. This is not a weakness of the science. It is science being honest about what it can and cannot claim. The strength of the Denisovan story lies precisely in this transparency. The genome data point to clear, statistically unambiguous signals of multiple archaic admixture events, and the fossil evidence, though fragmentary, is consistent with the genomic picture. More fossils will emerge in the coming years, and some will likely force revision of details. But the broad architecture of the finding is as solid as any result in modern population genetics. Papuans carry archaic human DNA at levels unmatched anywhere else on Earth. That DNA came from at least 2 different archaic populations. And those populations were as genetically divergent from one another as Neanderthals are from us. Let that sink in one more time. The entire Denisovan portion of the story was hidden, invisible, for the entire history of scientific inquiry, until a single finger bone met a sequencer in a clean room in Leipzig in 2010. Now the final twist, the one that connects everything to the present and to a much broader human truth. Because the Denisovan story is not just about Papua, it is about what it means to be modern human in the first place. For most of the 20th century, the prevailing model of our origins held that Homo sapiens arose in Africa, spread outward, and replaced all other human groups without admixture. The phrase was, "out of Africa with replacement." The symbolic picture was a wave sweeping clean across the continents, erasing every archaic population in its path, leaving only us. That model was clean. It was also wrong. The Neanderthal genome, sequenced by Pabo's same team just before the Denisovan breakthrough, showed that all non-African modern humans carry between 1 and 4% Neanderthal ancestry. Africans carry very little, though recent studies have shown that some sub-Saharan African populations carry traces of archaic admixture from as yet unidentified ghost lineages within Africa itself. This means that the Denisovan admixture in Papuans is not a weird outlier. It is the extreme end of a spectrum that includes all of us. Every modern human population on Earth carries the genetic signatures of ancient encounters. Interbreeding with archaic humans was not a rare exception to the rule. It was the norm. Modern humans moving into new environments, facing new diseases, new climates, new selection pressures, repeatedly absorbed genetic material from the populations that had already been there for hundreds of thousands of years. The Denisovan story in Papua is simply the most dramatic example we have discovered so far, the one where the archaic signal is loudest and the archaic sources are most diverse. This reframes what a Papuan genome actually represents. It is not just the personal ancestry of an individual from a specific region. It is a layered record of human survival, adaptation, and encounters stretching back 500,000 years. The Denisovan D1 and D2 populations had histories of their own, complex evolutionary paths through Southeast Asia that we know almost nothing about, biological adaptations and cultural traditions that are now represented only in the statistical whispers detected by genome-wide association studies. When a Papuan Highlander walks through a mountain garden, the genetic instructions building their cells, fighting their infections, processing their foods, come in part from archaic populations whose faces we will likely never see, whose languages will never be reconstructed, whose names we will never learn. They are not extinct, they are incorporated. And the communities that carry this legacy are not museum pieces. Papua New Guinea is home to roughly 840 languages, the highest linguistic diversity of any single polity on Earth. The highlands alone contain dozens of ethno-linguistic groups, the Enga, the Huli, the Tairora, the Kalam, each with deep oral traditions and cultural histories that have evolved continuously over those same tens of thousands of years. The DNA is not separate from that story. It is the deep chronological layer beneath it, the biological parchment on which the later chapters got written. If you carry roots from this corner of the world, some version of this archaic human history is literally sitting in your genome right now, in the sequences between the genes, in the regulatory regions, in the conserved immune cassettes, in the metabolic pathways. It is expressing. It is doing what genes do. The Denisovan pulse is not just a fact about the past. It is a fact about the body of anyone from that lineage. The textbook picture of human origins is still catching up to this. School curricula in most countries still teach a linear, replacement-based model. The public understanding of human evolution still features a succession of forms marching in single file toward a modern endpoint. The Denisovan discovery, the multiple lineage finding in Papuans, the functional adaptive introgression demonstrated in Tibetans and now in Melanesians. All of this constitutes a revision deeper than any since the mid-20th century. Human evolution is not a tree with a single trunk. It is a reticulated network, a braided stream, full of splits and rejoinings, a labyrinth of populations meeting and mixing and moving on. History is not finished. DNA has rewritten a chapter that the textbooks, even the most recent ones, are still scrambling to keep up with. The Denisovan story is genuinely one of the great scientific revelations of our time, and it is unfolding right now, in real time, in papers published in Nature and Cell and Science and Current Biology, by scientists who know that the next genome they sequence might contain another ghost lineage nobody has seen yet, another archaic pulse hidden behind the noise, another branch of the human family that has been waiting, silent, inside the cells of a living community somewhere on Earth. The next finding could easily come from the highlands again, because Papua is not a place at the end of the world. It is the place where the deepest section of the human epic is still being read. Subscribe. We go this deep every time, past the comfortable version of the story, and into what the DNA actually shows.